I'm studying Syntrichia caninervis and their skewed sex ratios, and I am analyzing them genetically by developing microsatellite markers. And so this desert bryophyte, it reduces expression of sexual organs in times of increased temperature. And you'll find really odd sex ratios with lower elevations where it's hotter, having, when they do express sex, they will just be female, and you usually won't find any male individuals at the lower elevations. And then at the higher elevations where it's a little bit cooler, you'll have populations. When they do express sex, they'll obviously be more female, but you will find a few males. In a previous study at a higher elevation, one population had 14 females and just one male. And so a lot of research has been done on this before, but we are analyzing it genetically. And step one was to create microsatellite markers in order to be able to assess the population. And so primers were developed for this species, and we had 64 primer pairs that were tested. Out of those, we had 30 amplifiable loci. And out of those, we got 14 microsatellite markers that showed variability. And so here are an example of 3 of the microsatellite markers. And these were for 4 different individuals from across the Mojave Desert. And this is just an example, but each one of these shows variability. So already we are seeing that already in individuals that are from the same population, that they are not from the same plant, and that they're variable between individuals. So what we're trying to do is look at the whole population and see, are they producing clonally or are they outcrossing? Because if they're not expressing sexual organs and we don't have any males, and they are dioecious species, so there's a male and a female, and are they reproducing sexually or are they reproducing clonally? And if they're reproducing clonally, how are these populations being sustained on clonal growth alone? So the development of the microsatellite markers is going to be able to help us go back and sample the whole population and see which ones are clones and which ones have been outcrossed. And so our next step is to go back this summer and sample across the Mojave and do multiplex PCR. And then we'll be able to see how these populations are sustaining themselves, if they are having sex or if they're sustaining themselves clonally.